My name is Joshua Mugabi. I welcome you to Topic 2, Nutrition in Plant. Our learning outcome, A. Evaluate the relationship between the structure of chloroplast and photosynthesis in C3 and C4 plant. Details of biochemistry are known to required. Learning activity. A. In groups, learners study images, tactile diagrams of the ultrastructure of a chloroplast. Make a model and use it to explain the functions of the parts. Let's start with the ultrastructure of a chloroplast and functions of its part. Item 1. We shall start by observing chloroplast ultrastructure. Students are shown an electron micrograph of chloroplast from a leaf mesophyll cell. They are required to identify the internal structures and relate them to photosynthesis. Task: identify the chloroplast as seen in the micrograph. Name 6 ultrastructure components of the chloroplast. State the role or the function of each named part. Identify the chloroplast as seen in the micrograph. The organelle is a chloroplast. Name 6 ultrastructure components of the chloroplast. There is the outer membrane, inner membrane, intermediate intermembrane space, the stroma, thylakoids, grana, lamellae, chloroplast DNA, and 70S ribosomes. State the function of each named part. We have the ultrastructure part and the function. Outer membrane. This controls movement of substances into and out of the chloroplast. The inner membrane encloses the stroma and regulates internal environment. Intermembrane space separates the two membranes. The stroma contain enzymes for light-independent reaction, the Calvin cycle. The thylakoids, these contain chlorophyll. They are at the site of light-dependent reactions. We have the grana. This increases surface area for light absorption. The lamellae, they connect grana for efficient transfer of electrons. Chloroplast DNA. This codes for some chloroplast proteins. 70S ribosomes. They are for protein synthesis within the chloroplast. Starch grains. They are temporary storage of photosynthetic products. Let's now look at the ultrastructure of chloroplast and functions of its part, item 2. We are going to be relating structure to photosynthesis. A learner asks why chloroplasts have many stalked membranes instead of a single internal membrane. Task: identify the membrane structure referred to. Explain how their arrangement increases photosynthetic efficiency. Identify the membrane structure referred to. They are the thylakoid membranes arranged into grana. Explain how their arrangement increases photosynthetic efficiency. Stacking increases surface area for chlorophyll. More light energy can be absorbed. This allows more photosystems and electron carriers to function simultaneously. We are still under ultrastructure of a chloroplast and functions of its part, item 3. We are going to compare chloroplast and mitochondrial ultrastructure. Students, compare chloroplasts and mitochondria under an electron microscope. Task: state 2 similarity in ultrastructure. State 2 differences in ultrastructure. Explain the biological significance of one similarity. State 2 similarities in ultrastructure. Similarities: both have double membranes. Both contain their own DNA and ribosomes. State 2 differences in ultrastructure. Differences: chloroplasts have thylakoids and grana. Mitochondria have cristae. Chloroplasts contain chlorophyll. Mitochondria do not. Explain the biological significance of one similarity. Presence of DNA and ribosomes allow partial self-replication and protein synthesis. Still under ultrastructure of a chloroplast and functions of its parts, item 4. We are going to look at the function consequences of damage. A herbicide damages the thylakoid membranes of chloroplasts in crop plants. Task: identify the process directly affected. Explain the effect on the overall rate of photosynthesis. State one visible effect on the plant. Identify the process directly affected. Light-dependent reaction. Explain the effect on the overall rate of photosynthesis. ATP and NADPH production is reduced. Light-independent reactions slow down. Overall photosynthesis decreases. State one visible effect on the plant. There is reduced growth, yellowing of leaves, which is chlorolysis, and reduced yield. Marking emphasis. Correctly identify the ultrastructure parts. Actually state functions. Relate structure to photosynthetic processes. Use correct biological terminology and present responses in clear logical points. Conclusion. The chloroplast's ultrastructure is highly specialized for photosynthesis, with which components contributing to efficient light absorption, energy conversion, and carbohydrate synthesis. Thank you for participating in topic 2, nutrition in plants. Our learning outcome A, evaluate the relationship between the structure of chloroplast and photosynthesis in C3 and C4 plants. Details of biochemistry are not required. And our learning activity has been A, in groups, learners study images, tactile diagrams of the ultrastructure of chloroplast. They make model and use it to explain the functions of the parts. If this has been very useful, please do visit me at Mugabi Joshua Biology on YouTube.